Chapter 7, Section 4, Transcription Controls in Archaea. Archaea transcription regulation resembles what we see in bacteria and what we see in us eukaryotes. Okay, that's the use of these transcription factors, especially here in eukaryotes. Archaea use both the positive and the negative. Okay, allosteric activation of an enzyme means it's going to go and recruit in polymerase better, or activation of a repressor means it's going to bind and interfere with polymerase. We see that repressor proteins in Archaea either block RNA polymerase or they block the proteins that should be recruiting and aiding in the positioning of RNA polymerase. Some of these proteins that should be there, TATA binding protein, TBP, TATA, hopefully you remember this for intracellular genetics, maybe cell biology, I think Albie covers it. TATA boxes are sequences in promoter regions that are AT rich, TA, 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 hence TATA. And then transcription factor B. So these repressor proteins can interfere with these, which would, if they were bound, activated and bound to DNA, would recruit in RNA polymerase. So if they're not there, well, then it's a small chance that RNA polymerase will bind at the promoter and begin expression of a gene or an operon. You take an example, a repressor protein known as NRPR from Methanococcus maripaludis. We're just going to call it Methanococcus. Okay, repressor genes bind, interfere with expression of genes responsible for nitrogen assimilation or nitrogen fixation. Alpha-ketoglutarate levels rise. Well, this is where you're going to get your ammonia, your nitrogen in Archaea. Okay, you're going to strip it out of amino acids. If ammonia is limiting, if it's high, it's low. If the alpha-ketoglutarate levels are high, if they're low. Here's our two counter signals that are going to control if and when NRPR is activated. Alpha-ketoglutarate is the inducer. Alpha-ketoglutarate, under normal conditions, NRPR is there. It's bound. It will not allow the positioning of RNA polymerase because it's bound at the TATA box. It's interfering with, TFB and TBP, two transcription factors that aid in the recruitment and positioning. Alpha-ketoglutarate binds and pulls away. Alpha-ketoglutarate in high levels of ammonia becomes glutamate. Well, boom. If low levels of alpha-ketoglutarate, NRPR levels active. No reason for nitrogen metabolism enzymes. But ammonia is there because levels is high. It's going to bind alpha-ketoglutarate, or I'm sorry, these levels are low. Then you have lots of alpha-ketoglutarate. This gets bound, allosterically inhibited. You get expression. High levels of ammonia means that this bacteria has done all the fixing of nitrogen it needs to. Well, alpha-ketoglutarate becomes glutamate, shut everything down. No reason to keep making the enzymes that are responsible for nitrogen fixation. We see, though, that many of the transcription factors, the regulators in Archaea, actually are dual purpose. They can act as both repressor and activators depending upon co-signals, depending upon the availability of certain repressors, certain co-repressors, co-activators, and whatnot. Example of this is the protein TRMBL1. It represses other sugar transport system, activates genes for glucose synthesis, also known as gluconeogenesis. Under normal conditions, TMBL1 binds to, interferes with, boom, the genes for sugar uptake. Okay? Because if there's no sugar out there to be taken up, there's no reason to go through and do all that. Well, look, maltose gets in because it's suddenly in the environment. It now diffuses across the membranes in the cytoplasm. Maltose binds TRMBL1, interferes with, pulls it off. These other two, TBP, TFP, recruit in position, RNA polymerase, boom, expression of that operon. Are there other conditions, other genes? Well, guess what? TRMBL1's binding is binding upstream, and it's recruiting in all those other transcription factors and all those other proteins that is going to now allow for polymerase to be bound and positioned correctly. You get expression of the genes for glucose synthesis, gluconeogenesis. Get enough sugars being made, or you got enough sugars there. You don't need to keep reversing glycolysis, taking your pyruvates and turning them back into G6P. You got other sugars that can be modified. Well, it now binds to, pulls off TRMBL1, which pulls off the other proteins, which also pulls off RNA polymerase. No expression of that operon. Here it interferes, here it aids, and its lack of being there interferes with further expression.